Hi there, and welcome to our 5-minute dive series of Proteins Unfolded, the podcast where we explore the extraordinary world of proteins and how they shape life as we know it. I'm Andrea.
And I'm Emilio.
And we are researchers at the Human Protein Atlas, based at SciLifeLab. In today's episode, we are not going to talk about proteins for a change. Instead, we'll explore something that's intricately and closely related, the transcriptome.
With us today we have Evelina Sjöstedt, in charge of the cell type research within the Human Protein Atlas, a resource with a large focus on transcriptomics. Thank you for being here.
Hi, thank you for having me.
Why don't we start by answering the question at hand? In our first episode, we talked about the proteome as a complete set of proteins being made in our body or in a specific part of it at any given moment. We also mentioned how the proteome constantly changes, unlike the genome, which virtually doesn't change throughout life and remains constant. Evelina, how would you describe the transcriptome in this frame?
Well, the way we at the HPA see it is that if proteins are the final products, the ones doing the work, transcripts are like the instructions from the DNA. The transcriptome is the full set of RNA molecules that are currently present in a cell. It's like a real-time printout of what the body is currently doing or planning to do.
Would you say then that the transcriptome tells us which genes, quote-unquote, are being read at a specific time?
Exactly. DNA is stored deep inside our cell's nucleus, but it can't leave. So when your cell needs a protein, it turns the instructions from DNA into RNA, which is a mobile version of that information. This mobile message is what gets translated into a protein in the cell cytoplasm. So by looking at the transcriptome, we're seeing the set of instructions actively being used at a particular moment.
And just like the proteome, the transcriptome changes depending on what's going on in our body, right?
Yes, it's highly dynamic. It changes with things like stress, diet, sleep, infections, you name it. It's context dependent. For example, immune cells in your body may express different transcripts when you're sick compared to when you're healthy. The transcriptome tells us what the cell is preparing for, what's being prioritized, which means you can pretty much ask the same questions as when mapping and measuring proteins.
Thank you. So now that we have the basics, let's dive in a little more deeply. Evelina, one of your specialties is a relatively new field of single-cell transcriptomics. Would you tell us what exactly this means?
Well, as you said, it's a relatively new technique where we can see not only which transcript are present, but in which individual cell. Normally, when we sequence DNA or RNA, we will extract this material from a sample and get an overview of what's happening in the sample as a whole. But imagine being able to see which genes are being read in a specific cell of a mouse brain or in a tumor section compared to other cells. That's what a single-cell transcriptomics does. It combines cell sorting technologies with RNA sequencing to map gene activity for each individual cell.
So you don't just know what is being said, but where it's being said inside the tissue.
Exactly. This is helping us to understand complex tissues like the brain in much more detail.
Quick question. If we already have transcriptomics, why do we still need to study the proteome?
Great point. In a way, RNA is a proxy. It tells us what might be made, but not all RNAs become proteins. And in fact, it gets more complicated than that because RNA molecules can also be actors themselves. Some have functions beyond being messages between DNA and protein. When it comes to proteins, the transcriptome gives us one part of the story, while the proteome, more directly, is what's actually there. However, from a technical perspective, studying the transcriptome or the proteome has different advantages and disadvantages. So it's important to see the full picture and what the transcriptome is telling us as well. In the end, the proteome and the transcriptome complement each other, especially when it comes to understanding the full picture.
So to wrap this up, we wanted to ask you what you find the most exciting about studying the transcriptome.
For me, it's the ability to see the starting point, what a cell or tissue has potential of doing. It's like seeing their toolbox. Yes, the protein is doing more of the actual work, but the completeness of transcriptomics is the ability to get a better overview.
So, to sum it up, the transcriptome is a real-time voice of the genome. It's what your cells are saying they're going to do, like a to-do list for protein production.
And together with the proteome, it gives us a window into the current state of your body, helping researchers like us to understand health and disease in a much deeper level.
Evelina, thank you so much for joining us today.
Thank you.
And thank you for tuning in to Proteins Unfolded. If you enjoyed this episode, Share it with someone curious about how their body works, or somebody who loves a good science story. And remember, if you have a question you'd like to ask us, send us a message.
See you next time!